It's a pleasure to be here today at NIMBioS giving this talk. And today I'm going to talk about some of my postdoctoral work at the University of Georgia. Actually, the bulk of my talk will be spent developing a theoretical framework for forecasting epidemiological transitions. That framework, I will use it to derive leading indicators or early warning signals of epidemiological transitions, or these critical transitions in infectious disease dynamics. And then I will talk about a simulation study that I conducted to assess the performance of these early warning signals and data. And all of this will form the foundation for my proposed work here at NIMBioS. So what is an epidemiological transition? Infectious disease emergence can be considered a critical transition because it involves a transition from stuttering chains of transmission to sustained chains of transmission. So in other words, the disease becomes endemic. And usually when a disease emerges, there is a major rise in infected cases. And this may be due to the introduction of a novel pathogen, or it might be a re-emerging pathogen that has been present for a long time, but due to long-term changes in the environment, or in the host behavior, or in the pathogen itself, the disease may end up re-emerging. On the other hand, infectious disease elimination is also considered a critical transition. And this is where we have the transition where the disease is endemic, but a tipping point is crossed, and cases begin to decline until there is no more infected people left, and the pathogen becomes extinct. This is also considered a transition. And the inspiration behind some of these ideas came from this really interesting paper by Lloyd Smith et al. In 2009. And in this paper, they talked about the phases of emergence for zoonotic pathogens, a zoonosis being a pathogen that can be transmitted from animals to humans. And what they talked about was the process of spillover from an animal to a human. And the pathogen may then adapt to the human host. But before it adapts and before transmission is readily sustained, outbreaks may die out, or the infection may die out. But due to regular importation of the pathogen, regular spillover events, or due to evolution, or due to other slow changes, say for example in recruitment, the transition to endemic disease may eventually occur. So what might this look like in case reports? Here I'm going to show you Pertussis case reports in California. Pertussis is a childhood immunizing infection that in the 1940s, a vaccination program was rolled out in the United States to control it. And by the 1970s, cases had reduced to almost nothing in most states. But from the mid-1970s onwards, there was a very slow resurgence in cases, before eventually preceding a major outbreak in 2005 here in California. And so we asked the question, could this have been anticipated before it occurred? This is just one example, but another example is malaria. And here you can see a time series of case reports of malaria cases in Kenya. So in the 1940s and 1950s, there was a global eradication campaign in malaria, which almost succeeded. But the eradication campaign was stopped in 1969. And during that campaign, in some countries, the disease was eliminated. But in other countries, such as Kenya, it was not. And eventually, over time, the disease eventually resurged in Kenya and in other countries too. And this may have been due to changes in climate or changes in land use. It's difficult to know here. But the key idea that I want to talk about today is that this transition may have been due to non-stationarities. In other words, epidemiological processes that are slowly changing through time, slowly and gradually and smoothly. And this is one hypothesis for this type of transition. Here I'm just showing you a schematic of a driver that may be changing gradually and smoothly through time. And these drivers may take different forms. It may be evolution of the pathogen. It may be the rollout, it may be a decline in vaccination practices, which is one thing that I'm going to be talking a lot about today. It could be changes in birth rate, as may have happened for pertussis. Or it may have been these changes in land use or changes in agricultural practices, or even changes in human behavior. So there's lots of different potential drivers of these changes. But can we actually build these drivers into mathematical models? And I'm going to introduce a key idea from epidemiological theory now, and that is the basic reproduction number. And the basic reproduction number is the number of secondary cases generated by a primary case in an entirely susceptible population. But the key idea about it is that it is a critical threshold or a tipping point, in that when R0 is less than 1, small outbreaks, there are small outbreaks with occasional direct transmission, but the disease will eventually die out. On the other hand, when R0 is greater than 1, the disease will persist, or the disease is endemic and there is sustained direct transmission. So it makes us think about bifurcation theory as a natural conceptual framework for looking at this in more detail. So now I'm going to introduce the SIR model with demography and vaccination. So this is a very simple epidemiological model commonly used for childhood immunizing diseases such as measles and pertussis. And here I've just going to talk about the assumptions of the model. Recruitment into the population is given by this term here, mu times 1 minus p. We assume that transmission is given by this term, beta times S times I. Population size is assumed to be constant, so the mortality equals the recruitment rates. Finally, infectious individuals can recover and become permanently immune at per capita rate gamma. The key parameter that I want to draw your attention to here is the parameter P. And this is the vaccination uptake. So vaccination uptake here being the fraction of the population that's vaccinated. And this, we will consider this to be our bifurcation parameter, in that this parameter we assume to be slowly changing through time. But before I talk about that, I want to introduce the bifurcation diagram for this system. So this system has two equilibria, a disease-free equilibrium where the infectious population is 0, and an endemic equilibrium where the infectious population is positive. The basic reproduction number for this system depends on the fraction of people vaccinated P. So as the fraction of people vaccinated in the population increases, R0 reduces eventually to 1, where that is the threshold for elimination here. So in this diagram, I'm showing you the infectious population, and as P increases, eventually the transcritical bifurcation is reached. And this is where the endemic equilibrium and the disease-free equilibrium, represented by this red branch here, so these are the branch of stable equilibrium points, and these are the branch of unstable equilibrium points, they meet and exchange stability at this point. On the other hand, if a disease is emerging, it is the disease-free equilibrium that is stable, represented by this black branch. And as P decreases to the bifurcation point, eventually, again, these equilibrium meet at the critical point, and so the disease emerges. But the key question we want to ask, is this bifurcation actually detectable? And the answer is yes, if there is critical slowing down in the system. And critical slowing down is where the recovery rate from perturbations to an equilibrium becomes slower and slower through time. And the essential idea is that critical slowing down is actually based on the eigenvalues of the system. I will be talking a lot about eigenvalues in this talk. But these change, and at the bifurcation point, the dominant eigenvalue is 0. So the dominant eigenvalue representing the return rate gets progressively smaller as the bifurcation is approached. The interesting thing is that these may actually offer model-independent diagnostics of the proximity of a critical point, in this case, emergence or elimination. So I'm going to talk about the second part of inspiration in this idea, and that is the idea of fold bifurcations. So I've already talked about transcritical bifurcations, but this idea was first applied in ecosystem models and other complex systems where systems were actually bistable. So when I say bistable, it means that there's two alternative stable states. And these alternative stable states are represented by these stability diagrams here. And the equilibrium point is the minima of this potential function. So that we can see there's two minima here. And you can see from this ball and landscape diagram that if we imagine that if the ball is pushed off this, this minimum point here, so representing a perturbation, because the slope is steep, the ball would roll rapidly to equilibrium. And that is the idea of the eigenvalue, that is, the return rate is fast. So the system has high resilience. On the other hand, if the potential function looks like this, and that the landscape is now shallow rather than steep, you'd expect that when the ball is pushed off its equilibrium point, it would take longer to return to the equilibrium. And I'm just showing you here a fold bifurcation diagram. And a fold bifurcation is when a small and sudden change in the parameter leads to a catastrophic collapse in the population, as you can see here. So this branch of points, at this critical point, the population suddenly drops down to this point when you just shift the conditions just a tiny bit. And you can see that far away from the bifurcation point, for these equilibria, the system is highly resilient and there are small fluctuations. On the other hand, if we're close to the bifurcation point, the system has low resilience and there's large fluctuations. So the key idea I'd like you to take home here is that these fluctuations may actually contain statistical signatures of this critical slowing down, and that there may be generic warning signals for this for the approach to the tipping point. For example, there may be an increase in variance or skewness, represented by the fluctuations here getting larger, or because the memory in the system is increasing, in that because of the return rate getting slower, points in time are more and more like their past state. That may be indicated by an increase in autocorrelation in the system. So critical slowing down has been detected in a number of natural systems. And one of the first cases was this was the first experiment where critical slowing down was detected. And in this experiment, Drake and Griffin, they had a Daphnia and Macdonald population. And that population, they were exposed to less and less food through time. And they were compared to a control population, which were given a constant amount of food through time. And various leading indicators were measured. Here I'm just showing the coefficient of variation, which was shown to increase as the tipping point was approached. Detection of critical slowing down has also been found in ecosystems, where Carpenter et al. Had a natural experiment where they had a control lake and a lake where a predator was slowly introduced to the lake, and thus causing the prey population to slowly collapse. And again, they looked at various early warning signals of the collapse, and they found that it could be indeed detected. Critical slowing down has also been detected in medical systems, including physiological systems. Epileptic seizures are the determination of epileptic seizures is difficult to detect. And in this experiment, the authors had data on electrical brain signals, and they looked at the power spectrum and temporal correlation, spatial correlation, and they were all found to, in these cases, these increased, but the power decreased towards lower frequencies. All of these indicating that critical slowing down was occurring. And in that situation, they had a subcritical, they assumed it was a subcritical half bifurcation that was causing the transition. Finally, critical slowing down has also been found to precede climatic transitions. In this paper, they looked at various climatic transitions, including the greenhouse icehouse transition, and found that the autocorrelation increased in each case. So it's been documented in lots of systems. But can it be detected in epidemiological systems? And we think that it can, because at the critical threshold of R0 equal to 1, the dominant eigenvalue is equal to 0 of the system. And so the characteristic return time would diverge. So just to summarize here, I've shown that what epidemiological transitions are, and that critical slowing down has been detected in lots of systems. Now, can we apply these ideas? And so that involves developing a theoretical framework to answer the following questions. How might critical phenomena manifest in infectious disease systems? Can that transition be detected? And do time series data actually contain these statistical signatures that characterize the approach to a tipping point? So before I go on, I'd like to acknowledge my collaborator at the University of Georgia, John Drake. And he did a lot of foundational work of detecting transitions in ecological and epidemiological systems. It was also his idea to play a game of Jenga at one of our lab meetings. And we basically filmed ourselves playing the game. And it's a nice demonstration of a tipping point. Because for those of you not familiar with Jenga, it involves building a tower of blocks. And what you do is you take out the blocks one at a time and stack them on top of each other. And so the system becomes, as the blocks are moved up to the top, the system becomes more and more unstable before the blocks eventually collapse. So the object of the game is to avoid that happening, because if you take out a block and it falls, you're the loser. But what we did find is that as you stack up the tower, that the stability becomes less and less, in that it takes a little bit longer each time to return to its steady state of standing. So before to move on to the next part of the talk, I'm going to introduce the SIR model with immigration. And so if you recall before, I talked about the SIR model without immigration. This is the same model, except here I have this immigration term. And you can recover the model I talked about before by setting the per capita immigration rate, eta, to 0. Basically, all this represents is an importation of infectives from outside sources, perhaps just due to people leaving the closed system and picking up the infection from elsewhere, or from sick visitors visiting. And so in this diagram, I'm just comparing the equilibria of the systems with and without immigration. So the black line represents with immigration, and the red dashed line here represents without immigration. So this system behaves very similarly to the last one. But anyway, what I want to just make the point here is this system has a single equilibrium point. And for those of you with knowledge of nonlinear dynamical systems theory, that equilibrium point can either be a spiral or a stable node. And this, it's a good limit case system of the system with the transcritical bifurcation. So this model actually does not have a transcritical bifurcation, because close to the critical point, represented by the threshold value here, P, there is generally close agreement, except maybe very close to it. But it acts like because this model is a little bit more realistic and allows for a non-zero infectious population, we decided to use this. And so to model the fact that vaccination uptake can change slowly and gradually through time, wrote down a fast-slow system of the system with immigration. And what I want to draw your attention to here is that the rate of change of vaccination uptake is changing slowly through time. And it's actually proportional to this parameter epsilon, which is a very small parameter. And what we're saying here is that the rate of change of vaccination uptake it's a very, very small relative to the other parameters and variables in the system. So everything else is changing quickly relative to the vaccination uptake. We assume that vaccination uptake change is actually linear in time. And when we set epsilon equal to 0, we recover the model with immigration, where the solution is the stable equilibrium. And I'm taking care to distinguish between these two cases because I will be talking about all of these models later in the talk. So the next thing I did was write down a stochastic version of this deterministic model. So here I'm writing down the changes in state and the corresponding transition probabilities which correspond to the rates in the deterministic model. So here this is a discrete process. And so we have infection and deaths and recoveries and recruitment. And we simulated both of these models, approaching elimination and emergence. So here I am showing the deterministic case, where this black branch represents the stable equilibrium points, and this limit case is, the blue case is the equilibrium points, and here is the black case is the solution of the fast-slow system for this value of P sub naught. But the point that I want to just show you here is that when we simulate the stochastic system, the demographic stochasticity, so I should have mentioned in the last slide that we're assuming that there's only demographic stochasticity in the system. So that is random fluctuations due to changes in births, deaths, emigration, immigration. This can excite the transient oscillations of that spiral that I was talking about in the deterministic system. And so we see these excitations as the elimination is approached. On the other hand, if we look at the system approaching emergence, what I want to draw your attention to here is that the solution of the fast-slow model is a little bit lower than the equilibrium, than the corresponding equilibrium at that time period. And so that delay in dynamics actually manifests when you look at stochastic simulations of that model. So here this dashed line represents where the critical point would be in the model without immigration. And you can see outbreaks are small and sporadic before the bifurcation point is crossed. But they remain small and sporadic up until maybe after 20 years after that point is crossed, before eventually there's oscillations. So I thought that that was interesting. And there is definitely some cases, like some discussion in the literature of how there can be long transients near critical points, and that these can take a long time to manifest. But also, if you look at stochastic theory as well, the probability of a major outbreak does depend on R0, at least for directly transmitted systems. And the probability of a major outbreak would remain small for maybe some considerable time after that bifurcation point is crossed. But still, I thought it was interesting. So to make sense of this, I did some mathematical work. So this is the slide that if I was a field biologist, I would have a lovely picture of me in the field, up to my neck in mud or something, with a pretty organism, but nobody took any pictures of me up to my elbows in math and paper and things. So, so here I go with this slide. So what I did was I took the stochastic process that I showed you, and I constructed master equations for the probability distribution of infected individuals. And to that, I applied the Van Kampen system size approximation. And that is basically, it assumes that that fluctuations scale with the square root of the population size. And it is nice because it allows us to go from discrete to continuous, which is an advantage here. Because from it, we get a set of equations for the deterministic system and a set of stochastic differential equations for the evolution of fluctuations through time. And it is from those stochastic differential equations that we can obtain leading indicator statistics. And that's the main advantage of going through all of these steps, because we can actually obtain analytical expressions for the summary statistics. And the ones that I derived were the power spectrum, autocorrelation, variance, and the coefficient of variation. And the main take-home I want you to take from this slide is that all of these expressions depend on the eigenvalues of the system, which, if you recall, is the return rate of perturbations from equilibrium. So we can then take these expressions and plug in parameter values. And the parameter values that I used were relevant for pertussis. I should have mentioned that pertussis is actually a very nice potential application for these ideas of forecasting epidemiological transitions, because measles and pertussis, both of them, the epidemiology is well known, and there's plentiful case report data. So we plugged in those parameter values. Yes. Mm-hmm. Yes. So I actually looked at both cases. I looked at eigenvalues for the Jacobian matrix, evaluated about the equilibrium, but also Jacobians evaluated about the solutions of the Vaslow system as well. Mm-hmm.
Is there a term in there that I can interpret population size?
So in the coefficient of variation, the population size pops up. Yes, and I don't recall exactly, but it probably pops up in eigenvalues as well. Yes, I didn't write down all of these expressions because they're fairly complicated. Okay, these are the terms of the covariance matrix in the set of stochastic differential equations. It is, I think, yes. Yes, it is, yeah. So, I also did the same analysis for the SIS model. In that model, you're just assuming that populations can, once they recover, they become immediately susceptible. But the expressions are much, much simpler and are just because it's just one stochastic differential equation. And what I want to show you here is that, like, for example, the variance will increase. They all depend on the dominant eigenvalue. The variance will increase as this dominant eigenvalue approaches 0. And similarly, you can see the same for all of these expressions. That this is where the generic leading indicators idea comes from, of increasing variance, increasing autocorrelation coefficient, increasing coefficient of variation, and the power spectrum becoming increasingly dominated by low frequencies. And I'm going to make some comparisons with that as well. Okay, so I'm going to just show you some of the theoretical predictions. And what I want to show you here is the power spectrum, which basically is a function that plots that basically shows what frequencies dominate in a time series. So as elimination is approached, the peaks become higher, which shows that the low frequencies begin to dominate. So these where the peaks, the center of the peaks is basically the dominant frequency. And you can see that the frequencies become lower as the pathogen approaches extinction. On the other hand, if the pathogen is emerging, for example, the power spectrum is actually flat, like a white noise spectrum. And this corresponds to the small and sporadic outbreaks that I showed you previously, that are not really dominated by a single frequency. But as the transition from emerging to endemic occurs, you'd expect the small sporadic outbreaks to be replaced by outbreaks that are dominated by resonant frequencies or resonant oscillations. P again is the vaccination uptake. Yeah, fraction of people vaccinated, and it's our bifurcation parameter. And in the case of pertussis, that might represent less people becoming vaccinated over time. Yes. So I forgot to, thanks for pointing that out. This corresponds to the model without immigration, and this is the model with immigration. And in the model without immigration, because the infectious population is actually 0, at p equals 0.95 for this particular system anyway, there is no power spectrum. So this is... Yes. Yes, exactly. Okay, so to move on to some of the other leading indicator statistics. And here I'm actually, again, I'm comparing systems with and without immigration here. This is evaluated using the eigenvalues for the Jacobian evaluated at the equilibrium point. And what the main take home from this slide is that there is increasing autocorrelation, decrease in variance, and an increase in coefficient of variation. So while these two agree with standard expectations, the variance is predicted to decline, which is not really in agreement with standard expectations or these generic indicators. I also calculated these for the fast-slow systems, and I calculated them for different for 2 different orders of magnitude. And so for fast-slow systems where the p parameter is changing really slowly, there is very close agreement. But if p is changing a little bit more rapidly, there are some fluctuations in this variance. But the trends are in general the same. What about for the emerging case? Here we predict that autocorrelation will increase, variance will increase, but the coefficient of variation is predicted to decline. And this makes some sense here, in that if you think back to the power spectrum, we saw that it was flat for like a white noise spectrum, and it was representing these small sporadic outbreaks without any resonant oscillations. And so as the pathogen is actually emerging, we're getting that switch from these sporadic outbreaks to ones that are more resonant. And so the variation actually declines a bit. And I also compare these with the fast-slow systems, and the trends are the same. So, how robust are these theoretical predictions? To answer that question, whether these behaviors are actually detectable, we conducted a simulation study of 500 stochastic, SIR, fast-slow, and stationary epidemics. And in that, I detrended, for each simulation, I detrended to remove the influence of the mean. And then I did a moving window analysis, where statistics were calculated over a moving window half the length of the time series. This is what this picture is representing here. So we calculate the statistic, and for that simulation then we have a set of statistics, and say for example the statistics may look like this. And so we calculated Kendall's correlation coefficient to see what the trend in the statistics was. The trend over time, whether it's increasing. And we did this for each simulation so that we get a distribution of correlation coefficients. And we generated null and test distributions. So the test distributions are the systems approaching criticality, and the null distributions are those that are the stationary epidemics. So the idea to have like the ones approaching criticality and a stationary epidemic is can we actually distinguish these patterns from a system that's approaching criticality versus one that's not changing at all. And we use these distributions then in receiver operator characteristic analysis, where we calculated sensitivity, which is the true positive rate, and 1 minus specificity, which is the false positive rate. And so if there is perfect detection, you expect false positive rate to be low and true positives to be at 1 all the time. But if points are falling along this line, then there is no ability to detect a change. In other words, the distributions are completely overlapping one another, and so there's no ability to detect. So just to show you that idea, here the false positive rate is the integral of the distribution of tau under the stable system to the right of this line. And then we compare with the system approaching criticality in red here, which is the true positive rate, also to the right of that line. And so for each of these, we plot that point. And as I said, that if you can imagine these two are completely overlapping, they would everything would lie on this line. Whereas if they are completely different, everything would lie along this line. But usually the truth is somewhere in between. But it's a way of detecting how well how the performance of this algorithm. The other thing is that we calculated the area under the curve, which measures how different it is from or the magnitude of the difference away from this line. And so these are the results of our simulation study. Here are the moving window predictions for very slow elimination. And so what I did here is I calculated from all of those simulations, I calculated the median statistics using two different detrending ways. The main thing is that they agree. And then I calculated the 95% prediction intervals from those simulations. And you can see that the theoretical predictions in general hold up of increasing autocorrelation, decline in variance, and an increase in coefficient of variation. And then the ROC analysis shows us that the areas under the curve are high, indicating it is possible to distinguish between a stable system and one that is slowly approaching elimination, at least in these cases. What about moving window predictions if elimination is occurring more quickly? And here we can see that the trends are in general the same, but there's more variability here in the variance statistic. And the ROC analysis shows that the variance area under the curve indicates that it is less accurate in distinguishing between the stable system and the one approaching elimination, because it's close to this straight line. And we also looked at slow emergence in this SIR model, but in contrast to the elimination case. Yes? So that was actually a way of detrending that I didn't talk about. So we detrended in two different ways, just to compare those two different ways.
So what is the sampling approximation?
Yes.
And the simulation is represents the spread around.
Well, this is the median statistic, say, from all of my, like calculating for 500 simulations, calculate the median statistic, say median variance, and then just, yes, calculating the 95% prediction interval or the spread around that, yeah. What I'm trying to show here anyway is that there is a lot more spread in this case. And in contrast to the case for elimination, it is a lot more, the statistics perform much more poorly in detecting emergence. In that our areas under the curve indicate early warning signals can't distinguish between a stable system and one that's slowly emerging. And they're very close to these lines, except maybe in the case of variance here, which performs a little bit better than autocorrelation and coefficient of variation. That is the change in the driver. So that's so. Oh yes. In the case for elimination, I looked at 1/30 as well per year, and 1/500 per year. So the first case for elimination, I looked at 1/500 per year, and then yes, yes, yes. So lessons learned from our simulation study is that the approach to elimination is preceded by an increase in autocorrelation, decrease in variance, and increase in the coefficient of variation. But the approach to emergence was preceded by an increase in variance, but patterns in autocorrelation and coefficient of variation were nearly imperceptible, as we saw from those areas under the curves. So we could argue that there's reliable predictions for leading indicators of elimination, but not for emergence. And this is due to these maybe bifurcation delays and other far from equilibrium conditions that are prevalent. The other thing that I forgot to point out about the emergence simulations is that we were pretty conservative in that we only used the first 10 years of the simulations just up to before the critical point in the model without immigration. Whereas if we take into account the delay, perhaps if we'd used more points, we would have detected some changes in our statistics. But we decided to be conservative, just using the models to determine how many points we should take. So to conclude, we do have theoretical predictions, and the critical slowing down is detectable in simulations of fast, slow SIR epidemics. So what about non-directly transmitted systems? I became interested in applying this idea to vector-borne disease elimination. And the simplest way to do this is to use the Ross-Macdonald model for vector-borne diseases. And this model also undergoes a transcritical bifurcation at R0 equal to 1. And it also undergoes critical slowing down, in that the dominant eigenvalue is 0 at that bifurcation point. And what I did was I wrote down fast-slow systems corresponding to different control measures. Here I've written down the time-varying per capita biting rate due to application of bed nets. So this is one potential control measure that can control biting, the rate at which mosquitoes bite humans. And again, I just assumed a linear decline in that biting rate. And so I conducted a similar analysis of deriving stochastic differential equations, deriving statistics, and doing an ROC analysis. And these are the results for biting rate. And now we see an increase in autocorrelation, an increase in variance, an increase in coefficient of variation. And the areas under the curve indicate that we can indeed detect elimination before it occurs. But when we did the same analysis for reduction of an infectious period, which might be due to administration of drugs, or the prompt administration of drugs, we found that there is a decline in variance which is predicted, and here in the case of autocorrelation, the transition was not so well detected by autocorrelation, but well detected by variance and the coefficient of variation. So just to conclude that part of the talk, I've developed, this is the first steps in developing a framework for forecasting epidemiological transitions. And there is a lot of work to be done here. Just to conclude anyway, this part is that critical slowing down is detectable in epidemic systems. And so it may be hard to establish rules of thumb. Perhaps we can't always say it's going to be an increase in variance. Maybe in these higher dimensional systems that have lots of eigenvalues, the approach may be more difficult to detect, or it may be predictions may be system specific. But I think the fact that we can detect this critical slowing down is good. So all of these ideas, they've motivated me and inspired me to start thinking about how we might be able to apply the ideas of fast slow systems theory and critical slowing down to detecting epidemics that are due to gradual environmental changes. So a lot of environmental changes, such as climate change and changes in land use, occur over longer time scales relative to transmission. And so there has been a rise in emerging and re-emerging infectious diseases that are due to human-mediated ecosystem changes, such as increased agriculture, deforestation, urbanization, and climate change. And all of these may potentially modify transmission pathways. For example, changes in land use may modify contact patterns or change behaviors of hosts in the system. Whereas climate change could potentially lead to changes in immune responses in hosts and changes to host and pathogen physiology, including maybe altering infectiousness and virulence of a pathogen. So it'd be interesting to actually incorporate these changes into models explicitly using this framework. On the other hand, I think it's important that models recognize that propagation of infectious diseases is a multi-scale process, and that these environmental drivers may act over large scales, but due to spatial heterogeneities such as habitat and climate, the impacts of the driver will differ at these spatial scales. So I think it's important to write down models that actually unify these scales and to ask the question, how do these processes influence emergent disease dynamics on large scales? Clearly, these gradual changes. And so my ideas now are to use climate change and land use as paradigmatic examples of these gradual environmental changes, and to incorporate some of these changes in host and parasite traits into models, which will hopefully generate some testable predictions and maybe help us answer questions such as detectability transitions over multiple scales. I'd also be interested in actually using data and applying some of these models and fitting models to data. So if anybody has any interesting data sets, I'd like to know. With that, I'd like to thank John Drake and Andrew Park at the University of Georgia, all of my collaborators, including Jonathan Lilly, who helps out with the Vector-Borne Disease Project. He's a high school teacher. And everybody at the Drake and Park Labs at the University of Georgia, and our funding sources as well. So with that, thank you for your attention.